Photosynthesis is a process that occurs in cells with two reactions. The light reaction, which we're going to look at, uses light energy in order to generate two molecules that are high energy molecules. One of them is ATP, which is our currency in the cell, and the other is NADPH, and this molecule is used in a lot of biosynthetic reactions. These two molecules then are used in the Calvin cycle, or the dark reaction, in order to generate sugars. So photosynthesis can occur in bacterial cells such as cyanobacteria, and also in plant cells. So we're going to think of it in plant cells as occurring in the chloroplasts on our thylakoid membrane. So this here is our thylakoid membrane. And one side of that membrane is our stroma, which is up here, and the other side is our thylakoid space. Now, this membrane has proteins associated with it, and one of them is called photosystem II. So photosystem II was the second photosystem discovered, but it's the first photosystem in the light reactions. So this has a number of components, and it has a special portion that's our reaction center that accepts electrons, and it has two particular chlorophyll molecules, so a pair of them referred to as P680, and this refers to the wavelength where they best absorb light. And we also have other chlorophylls in here. Now remember, plants, of course, where they perform photosynthesis, have a green color, and that comes from the chlorophyll molecules, and these pigments are found within the thylakoid membranes. So here's our chlorophyll molecules in here. So our light comes in. So remember, light is a wavelength, but it behaves like a particle. So a photon comes in, and we have a chlorophyll molecules. The electrons within an electron in them goes to a higher energy state, and that state is passed between the little chlorophyll molecules till it reaches this particular special pair of P680. So they can also get the electrons can also directly get excited if the light hits them directly. We have another photosystem which works in a similar way. Photosystem 1 is this one, so it was the first one that was discovered, and a somewhat similar structure. So we have chlorophyll pigments within this, like this. Just draw a couple of them. We have again a special pair of chlorophyll molecules. They're called P700. And again we have a reaction center. And we have the same thing that occurs here, where light comes in and our electrons are excited into a higher energy state. That state is passed on between them. Eventually we get the electrons in the pair of P700 excited. And when that happens, in both of these, a pair of electrons are transferred, so they lose two separate electrons into the reaction center here. And when that occurs, they change to have a positive charge because they've lost negatively charged electrons. Where are those electrons replaced from? Well, in this case, we have P680. Once it's lost electrons, it's the strongest biological oxidizing agent known. And we have an enzyme that actually removes the electrons from water. So water is split, and we get 2 protons plus half a molecular oxygen. And in this process, our electrons are transferred across. They're transferred separately, and they replace the lost electrons from P680. Our electrons have moved to the reaction center and through the protein into the electron transport chain. Now the electron transport chain that follows photosystem 2 is actually comprised of 3 different parts, but I'm just going to refer to it here as the electron transport chain. And in that process, protons are transported across the membrane. And those protons are transported here. The electrons flow through the electron transport chain and into photosystem 1. By then, they're no longer at a higher energy state. Remember that light here excited our electrons. Electrons were lost from P700 into our reaction center, and we had this then positive charge. The electrons that have come through here can replace them, ready for the reaction to start again. Our electrons that moved into the reaction center come across. They enter another electron transport chain. Now this one is different. It's made up of a protein called ferredoxin, but I'm just going to say electron transport chain here. And then I'll continue our thylakoid membrane across a little bit more, because we have a really important protein here called ATP synthase. So ATP synthase is our protein that produces ATP. And you can see it's producing ATP here. And we have a movement of protons. They are coming across the membrane, and they're coming across here. I'll talk about that in a minute. Now, where do our electrons come to? Our electrons are actually moved into a protein called NADPH reductase and used to produce NADPH. And that also is made from, if I can fit it in, NADP plus. So let's trace our electrons again. Light excites the electrons. They move from our special chlorophyll molecules into the reaction center. Light comes into photosystem 1, moves our electrons into the reaction centers. We then have electrons coming from water to replace those that were lost here, and the reaction can continue. And they are then coming through to replace those lost in P700, through to the electron transport chain, and ultimately used to reduce NADP to NADPH. In that process, this electron transport chain here transports protons across the thylakoid membrane. So we have a high concentration of protons on one side. Those protons then flow through ATP synthase. So we have ADP plus inorganic phosphate being converted into ATP, which is our currency for the cells. So the two important molecules that are produced here are NADPH, which is used in biosynthetic reactions, so will be used in the Calvin cycle, and ATP, which will be used as well.